All right, so this is chapter 2, part 4. We're continuing on with the different types of important macromolecules or organic compounds. Proteins, a very diverse group of macromolecules. They also contain carbon, the carbon backbone, hydrogen, oxygen, but a different element they contain is a nitrogen. So look for nitrogen in the structural formulas for proteins. They are the most human of all the other organic compounds. Humans are made largely of proteins. Our body makes proteins. Proteins do the work. A lot of the work in our body is enzymes. Um, so we need carbohydrates to burn for fuels, lipids for structural support, energy storage, and hormones. And proteins do a lot of the work. This is a picture of an enzyme, protein in the body. Proteins, just like carbohydrates, which are built, and lipids that are built by smaller parts. Carbohydrate or proteins are also built by different smaller parts. The smaller building blocks of proteins are called amino acids. Here's some pictures of some different amino acids: glycine, cytosine, lysine, and tyrosine. In the human body, there's 20 different amino acids. And all of them have the same basic structure. As you can see here, and the different parts are colored. So the blue over here is the amino group that you'll see. It's the same in every amino acid. They also have a carboxyl group that is right over here, the C double bonded to an O with an OH. And then on one side they have just an H on them. And then the group that changes is called the R group. See how this in the light orange here, boxed, that is different for every amino acid. So to form a dipeptide, so two peptides put together, you have a dipeptide. You join two amino acids. So if my necklace here, each one of these little shells was an amino acid, two together would be a dipeptide. And they are joined together via a strong covalent bond, specifically called a peptide bond. And to join those amino acids together, if you look down here, glycine plus alanine produces a dipeptide plus water. So this reaction is called dehydration synthesis, like we've talked about, because to actually join them together to form this peptide bond, an OH and an H have to be kicked out, which then forms water. So dipeptide is two amino acids. A polypeptide, like this necklace would be a polypeptide, contains 10 to 2,000 amino acids. So polypeptides or proteins, large proteins, can vary in the amount of amino acids that they contain. There are 4 levels of what are called structural organization. The first level is the primary level. Let me just look down here. Yeah. It's simply the sequence of amino acids. So alanine, glycine, lysine, leucine, the order that they're in. The secondary is a little bit more complicated. It either gets folded or gets in a helical shape. It makes it a little bit more complicated. That's the secondary structure. The third structure, tertiary, is it gets a 3D shape to it, okay? So it just folds a little bit more. The amino acids even bond between other parts of the chain to make it a little bit more globular. And the quaternary structure, again, it does more folding, more bonding to make a full-formed protein, which is globular. The best way to describe it, looks like a glob. The resulting shape of this protein influences its ability to bind and recognize other molecules. And also this sequence and shape determines kind of functionality, what it does. If you have curly hair, the proteins in your hair have more of a kink to them. Straight hair, that protein shape is more straight. Denaturation is when a complex protein is broken down into its primary structure. It's denatured, it's broken apart, the bonds are broken, and it denatures it down to its primary structure. the protein can no longer do its functions once it's denatured. It loses its shape and its function. And denaturation is caused by a hostile environment, like, for example, high temperatures. If you were to get extremely sick or have a child someday that has a temperature that's increasing or around 105 degrees Fahrenheit, you're going to want to call or even take them into the ER because temperatures that high, they're scary. That the kid's not doing very well. First of all, they're going to act like they're out of it, going to be lethargic, which is scary to begin with. But if it gets even higher, it can actually fry proteins in your brain, denature them. So then you can understand what will happen. They won't work. And if the proteins that do the work in your body aren't functioning, you're going to possibly pass away. So here again is the picture of what I just did with this necklace, okay? So the primary structure is just simply the sequence of amino acids. Secondary is right here. Either it takes a helical structure, it's called an alpha helix, or a beta sheet. It's like if you take a sheet of paper and you fold it, and that is a helical sheet. Like I used to like to do this for fun with paper. Okay. I don't know why that keeps turning off. There you go. That's a beta sheet. And then it gets, for tertiary, it gets folded even further. The sides of the strand bond with each other, and this is the quaternary structure. So an example of a protein that gets denatured, something that I really like to make, is eggs. The protein in the egg white is called albumin. And when you fry that egg or you cook that egg white, you're actually denaturing that protein. You're breaking it down into its primary structure. And the high heat does that. And you know that it's changed structure because you can visually see it change. What does an egg white do when it is cooked? It turns color, turns to white. And you can't reverse that, can you? You can't make it back to clear. A specific type of a protein is an enzyme. Enzymes are a protein, a type of protein. You have many different enzymes in your body that do different things. Like in your stomach, for example, you have different enzymes that digest milk or digest proteins. If you don't have the enzyme that digests milk, guess what you are? You are lactose intolerant because you don't have that enzyme that digests it. So enzymes catalyze reactions, metabolic reactions in our body. They speed them up in all of your living cells. The substrate is the specific part that upon which an enzyme has its effect. So enzymes are specifically made to find a specific substrate, like a lock and a key. They are specific to each other. Enzymes, because they can be very potent when they're working, they are under a lot of cellular controls in the body. So they aren't turned on the whole time or off the whole time. Your body controls them. So how they speed up chemical reactions, how do enzymes act as a catalyst? So here's our molecules that we want to have a chemical reaction. So enzymes actually increase the frequency of collisions because when atoms collide, they chemically react. So enzymes will cause them to collide more. They will also lower the activation energy, and they'll properly orientate those molecules. All of those things help speed up the reaction. Nucleic acids are the fourth type of organic compound, and they are the building blocks of them are called nucleotides. Here's a nucleotide below. Every nucleotide contains a phosphate group, so nucleic acids have phosphorus. That's what makes them different from the other organic molecules. They also contain a sugar, a 5-carbon sugar, and a nitrogen base. And this base can vary. Just like the other ones, they contain carbon. You can see here's carbon ring, hydrogen, oxygen, and they also contain nitrogen. And what makes them different is they contain phosphorus. And these, of course, you remember from biology, are The principal molecules that are in your genetic code, the DNA, deoxyribonucleic acid, and ribonucleic acid. They contain or carry genetic information, both DNA and RNA, and they control the formation of the proteins, which is like we just got talking about. Your proteins do the work along with your cells. The proteins are in the cells, of course, and the DNA and RNA control that. DNA contains the master template, which is in your nucleus. RNA relays the instructions, takes a copy of it, and then starts making the proteins specifically that your body wants you to make. And so here is DNA. So we have nucleotide, 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 and the same on this side. They are joined together in a long chain. And in DNA, there's two chains that are joined together in the middle by hydrogen bonds. That's like the zipper, up and down. And I'm not gonna get too much into the details of the DNA and RNA structure because you did that into biology. This is just a review of what nucleotides are used for. This is RNA. RNA differs in that it's single-stranded. It also contains a different base, uracil, and a different sugar called ribose. That's what gives it its name. And there's actually 3 different types of RNA: mRNA, rRNA, and tRNA. Nucleic acids are also used to make ATP, adenosine triphosphate, which is the energy-carrying molecule. It's crucial to your body. Your body temporarily stores energy, also makes more energy on demand, or when your body metabolizes things, it produces ATP via cellular respiration. Breathe in air, your body combines it, breaks it down with glucose to produce ATP. There's an enzyme, okay, so again, proteins are coming in, that catalyzes the synthesis, so makes the ATP. It actually adds phosphate. So when ATP is made, it starts as ADP, adenosine diphosphate, which means 2, and an ATP synthase will add a phosphate, and it will then make ATP, produces ATP. So when you take in one glucose molecule, that's that simple sugar, and during aerobic and anaerobic respiration, cellular respiration, your body uses ATP synthase to produce 36 to 38 molecules of ATP. And this is the equation for how that ATPase works. And again, it's a combination of dehydration, synthesis reaction, and hydrolysis reactions. This is pretty cool. A human will use up to his or her body weight of ATP over the course of a day. That means that each ATP molecule is recycled. So when ATP is used, when you use that energy, it releases a phosphate right here that produces energy, okay? And then your body uses ATP synthase to join these two back together, which is dehydration synthesis reaction, to produce ATP. When they're separated, water comes in, breaks this up, breaks up that ATP to produce ADP plus phosphate. And that is the end of part 4.